Hi everybody, my name is Dale and my presentation today is titled An Invasive Weed Responds to Water Stress Across an Environmental Gradient. So who am I exactly? I should probably start by introducing myself. My name is Dale Perkins and I'm currently studying at James Cook University here in Cairns. I grew up in a small town called Tully, which actually holds the Australian record for the highest average annual rainfall in a populated area. So I'm sure many of you can imagine what kind of environment I grew up in. It was far from a desert. And in 2017, I enrolled in my undergraduate degree, majoring in zoology and ecology. So a few months ago, I finished my honours thesis, which is what this presentation will be about today. But before I tell you explicitly what my honours project was about, I should probably establish some context. Alright, so one of the ways that scientists are currently using invasive plant research is in the context of a space for time substitution, where current changes across geographic space are used as an analogy for progressive changes over time. And by studying how invasive species locally adapt across these geographic or abiotic climes, we can actually better understand the ways in which populations can adapt to climate change, giving researchers some degree of predictive power. Invasive plants specifically can evolve rapidly to adapt to different climates present in the regions that they invade. And because invasive species can evolve rapidly, it's not unusual to find rapid local adaptation along these environmental gradients. So predominantly researchers have been using common garden experiments to identify rapid adaptive evolution in plants, and that's precisely what I've done in my honours experiment here. There are, of course, limitations to the ways that plants can adapt, and this is due to evolutionary trade-offs. So all plants have to maximize their resource use via trade-offs between competing physiological demands, and invasive plants have shown a fairly remarkable ability to rapidly and efficiently maximize those trade-offs. In some cases, apparently even to overcome them. Invasive plants specifically, though, those that spread across latitudinal and, say, drought gradients, have repeatedly been used to investigate these trade-offs, usually in regards to something such as, say, flower time and plant size. By identifying these trade-offs, we can better understand what precisely will restrict a plant's capacity to adapt to certain changes in climatic conditions. So, what did I set out to do? Well, I used sicklepod, an invasive weed, as a model to assess the role of environmental stress on specific trait expressions. I also set out to understand how local adaptations of different populations across a precipitation gradient respond to environmental stress. And lastly, I set out to understand how rapidly evolving invasive plants deal with environmental changes such as those associated with climate change. So how did I do this? Well, my common garden experiment required that we have 9 sites where we took seed samples from. So those sites are indicated here with the flags all the way down to 9. And from each site, we took seeds from an individual plant. Up to 7 individual plants here per site. Each individual plant provided 1 seed that was given ample water and 1 seed that was given a water stress treatment. And doing this across 9 sites with 7 individuals, 2 seeds from each site, gave us a very large sample size to actually see, to compare between sites, between treatments, and all of that. Some photos of me in the field working hard. Here we can see the seedlings on the far left. In the middle there, this one here is just the leaves ground down to a powder so that we can do isotopic analysis. And here in the bottom photo down here, you can see a clear difference already in the height of the plants just between the treatments there. Alright, so what results did I come up with? Well, here's all the p-values. You can see that we've got significant difference between the treatments, significant difference across latitudes, temperature and precipitation, not necessarily a significant difference between treatment and latitude, or treatment and temperature, or treatment and precipitation. When I talk about latitude, temperature, and precipitation, I'm talking about the latitude of the site of origin, the temperature of the site of origin, and the precipitation of the site of origin, just to be clear. So yeah, we got a lot of statistically significant results. Let's have a look at some of them. So here's one of them. On the x-axis, we have the mean annual maximum temperature in degrees Celsius at the site of origin, ranging from 32 down to 26. And on the y-axis, we have leaf number. Now leaf number was used as a bit of a proxy for biomass because we couldn't actually kill the plants and dry them out and measure them. We needed them for further experiments. So here we can see the stressed plants in the orange are always measured consistently a little bit smaller. There's less leaves than those in the control treatment. And this is sort of what you would expect. This does conform with our results conform with expectations based on drought avoidance and drought endurance theory. Yeah. It's also well understood that plants will close their stomata in response to water stress as a way to reduce water loss, and in doing so, will actually reduce their ability to sequester carbon from the atmosphere, thus slowing their growth rates. So that sort of basically is another explanation for what we see here. Could be dropping their leaves or producing less leaves. And on this graph, we have the mean annual maximum temperature again, as site of origin on the x-axis. And on the y-axis this time we've got our delta 13C, which is our carbon isotope value. So carbon isotope discrimination is kind of a proxy for water use efficiency. The literature tells us that lower carbon isotope values often indicate ample water supply and lower water use efficiency, and basically a larger stomatal opening. And that allows more CO2 entry and fixation. So what we're looking at here, we can see that the plants that originate in cooler environments showed a greater carbon discrimination, carbon 13 isotope discrimination, than those of plants in the lower, hotter environments. This tells us that the plants from those environments, the cooler environments, are actually more sensitive to water stress than plants from the hotter environments. I am, of course, mindful of our time, and I don't want to go over my limit, so unfortunately I can't go over every variable in detail, but we did find a lot of similar trends when considering other site conditions such as evapotranspiration, which encompassed all of our variables, along with precipitation, etc. Here's just some graphics of that you can see there. The trends are very much alike. So altogether, what did we find? Well, there's plenty of variation, even among the control plants, right? Which were all subject to the same common garden conditions. And this isn't exactly a trivial finding, right? The presence of differentiated phenotypes is a key factor for invasive success, and no one has actually described this for this species yet in Australia. So these plants are able to inhabit a wide range of environments, and when subject to severe water stress, are still able to complete their life cycle. We also found evidence for the presence of ecotypes with different water use efficiencies in Australia. So our carbon isotope data suggests that plants from environments with low annual temperatures are more sensitive to water stress than plants from environments with high annual temperatures. And again, this is the first time that this finding has been made in Australia. This finding actually pairs nicely with some of the research done in the USA. The USA research confirmed the existence of cold-tolerant ecotypes there, but of course, here in the tropics, cold tolerance isn't so much of the focus. Drought tolerance is more important and is more what we're worried about here. So specifically in the context of global climate change, that's something we're focusing on, and that's why it's important that we know how these plants will respond to water stress. The importance of local adaptation for invasive species response to climate change. So we can actually predict now how cyclopod will respond to climate change and to future climates, right? So if precipitation levels decrease, we can expect the warmer ecotype of this species to perform better. And if it wasn't, well, let me say that again. If precipitation levels decrease, we can expect that the warmer ecotype of this species will perform better. It wasn't as sensitive to water stress, right? Inversely, if precipitation levels increase, we can expect the cooler ecotype to respond better. This kind of predictive power will allow for more accurate expansion models so that management can better estimate the range in which these invasive species can occupy. And of course, management is already currently doing that. This is not a graph or a model with my data. This is just an example of some of the modeling that's been done to map where cyclopod can occupy and where they are currently occupying. So this isn't the end. In the process of doing all of this work, we were prompted to discover some questions. So